So, guys, this is the basic structure of an antibody, which is a 150 kDa molecule. We also call it immunoglobulin. And these antibodies are expressed on the surface of B cells and later on secreted by the B cells as well. So, like any peptide, it has an amino terminus, which is here, and a carboxy terminus, which is here. And towards the amino terminus here, you see the red, boxes here. These are showing the antigen binding sites. This is the variable region, which is present both in the longer chain of an antibody and the shorter chain of an antibody. The light chain and the heavy chains that we call it. So these are uh, having a hint region here. Some of the antibodies like IgM and IgE do not have a hint region, but other antibodies classes, they have a hint region. And all these peptides or all these different domains are connected to each other by disulfide linkages. This is the basic structure. So this is the variable region or the Fab region where antigen binding sites are found. Uh, and now in today's lecture, we would discuss about the genetic basis of this variation, that how this variation is ensured, because there are lots of antigens, different types of antigens, and these antigens would always find a suitable, antibody molecule on the surface of a B cell inside our body whenever they infect us. So there should be a lot of variation while making these molecules or these antibodies inside our bodies. So this variation is ensured at genetic level, and we are going to discuss the this variation detail. You also have seen this picture where I had shown you that the light chain and the heavy chains, both of them have a variable region, VL for the variable region of the light chain, VH for the variable region of the heavy chain. And you see NH3 plus groups attached here. This is the amino terminus. And the disulfide linkages are also shown here. Now, this heavy chain, you see that there are different isotypes of the heavy chains. So an antibody can have, uh, one, for instance, isotypes of the light chain, kappa or lambda, and similar is the case with these isotypes, mu, gamma, alpha, epsilon. So these different isotypes of the heavy chain, these are found in different classes of antibodies. So these actually determine the classes of antibodies. For instance, in IgG or IgM or IgE, you would find different, isotypes of these heavy chains. So if we move ahead, I can show you here that this is IgG, IgM, IgD, and in every one of them is a different type of heavy chain. And the sugar molecules attached to these proteins are also different. These are glycosylated proteins. So the number of sugar molecules and the types are also different in case of different antibodies. Um, you see that in the case of IgG, there is a gamma isotype of the heavy chain. In the case of IgM, there is mu, and there is delta in the case of IgD, and there is alpha in the case of IgA. In the case of IgE, there is epsilon. So these, heavy chain isotypes actually determine, the type, the class of the antibody. And there's this, the number of these heavy chain domains is also different in different antibodies. That's what we have discussed before. But today, just briefly about the genetic basis of the variation, that how this variable site which binds with the antigen is, produced, and what happens actually at the genetic level to ensure this kind of variation in this region. So the DNA sequence encoding a variable region is assembled from 2 or 3 gene segments. Uh, what are these 2 or 3 segments? For instance, this is a lambda light chain locus shown at the top, and you would see here it is written V lambda 1, V lambda 2, L lambda 1, L lambda 2, V lambda up to 30, J lambda 1, C lambda 1. So this is the lambda light chain locus, which is present on chromosome 22. And in order to produce the lambda chain, which has 2 parts, one is the variable part and the other is the constant part. So this is actually produced by joining uh, sequences with each other, which are present on chromosome 22. So, what are these sequences? These are variable sequences, and the yellow boxes show the joining sequences. So, there are different segments of the joining, sequences that have to be combined with the variable sequences. And there are constant sequences that encode for the constant region of the lambda chain, and there are different constant region segments present on chromosome 22 at different locations, and these have to be combined with each other to make a single constant region. So the intervening sequences have to be removed, and L is for the leader sequences, leader 1, leader 2, and so on. So, uh, similar is the case with kappa light chain locus. It is also assembled in the same way. These kappa chain light chain locus is present on chromosome 2. You can see that one light chain locus, the lambda, and an antibody, I had shown you that the light chain can either be lambda or kappa. So the lambda, if it is a lambda light chain and an antibody, all the sequences which have to be assembled and to make a coding sequence for coding this lambda light chain locus, all of them are present on chromosome 22. But in case of kappa light chain, you can see that all these sequences are present on chromosome 2. And the sequences are the same similar type of sequences. There are leader sequences indicated by L1, L2, etc. There's V kappa 1, V kappa 2, V kappa 3, up to V kappa 35. It means that there are 35 V kappa gene segments that have to be assembled to make this variable region. By combining these 35 segments with joining sequences, which vary from 1 to 5. So 1, 2, 3, 4, 5 joining sequences, and 35 kappa variable kappa sequences have to be combined with each other along with the constant region or the constant sequences present on chromosome 2, and all these would be combined with each other by recombination, and then these would be transcribed to make mRNA, and that would be translated to make the kappa light chain, protein for making this particular antibody. Similarly, if you take the heavy chain, again, there are similar type of sequences, and in addition to the variable sequences, there are diversity sequences as well. Um, the heavy chain locus VH1 means variable heavy chain 1 segment, gene 1 segment. VH2 means variable heavy chain 2 segments. So these are different segments, all of them present on chromosome 14. How many different segments of variable regions have to be, combined with each other? There are about 40, variable segments uh, that are present on chromosome 14, and they have to be brought together in order to code for this variable region of the heavy chain. And there are diversity sequences from, about 23 diversity sequences, about six joining sequences, and about nine constant region sequences that have to be, recombined with each other in order to code for this heavy chain, peptide. And the heavy chain peptide and the light chain peptides, these are the peptides that are going to make an antibody. So, for instance, I can show you in this picture. Uh, uh, if you look at here, this is the light chain. On the right side is the heavy chain. And here you would see it's written here, the germline DNA. And the germline DNA, these sequences are in this, order, are in different positions. There are leader sequences, variable sequences, joining sequences, and constant sequences. I had shown you that in the previous slide, as you saw, that chromosome 22 or chromosome 2 have these gene segments, different gene segments for variable joining and constant region. And for the lambda and kappa, there are different chromosomes on which these segments are present, and these have to be joined with each other. So this is the germline DNA where they are, dispersed or present in different locations. And then these variable in joining sequences finally here have to be, a rearrangement of this genetic or germline DNA has to be carried out in order to make the joining and variable sequences, combine with each other. And these sequences, the constant region sequences, are also brought together. There are lots of gene segments at different positions, so they are also brought together. And then there you see that finally you would have a variable, the leader sequences followed by the variable in joining sequences, and then there is, intervening DNA or introns in between, and then there is a constant region sequences that are combined together. So in the case of heavy chain, you see that leader, variable, diversity, joining, and constant region. For instance, C mu is shown here, and MC sequences. These are all present at different locations on chromosome 14, for instance, as I show you. Then there is somatic recombination, and as a result of somatic recombinations, first of all, the diversity in joining sequences are joined together, as you see here. And when these sequences are joined together, then the constant region sequences, these sequences, the diversity in joining sequences are joined with the variable region sequences that are combined with each other. So in the final VD joint rearranged DNA, as a result of somatic recombination that is taking place, the variable diversity in joining sequences would be combined. And the constant region sequences and MC sequences, they're also combined. There is intronic DNA, which has to be removed once the peptide has to be synthesized. Or when mature mRNA is formed, then these sequences are removed and a transcript is made, and that is translated into a peptide. So these are different combinations of these sequences, and you can say that these different combinations can give rise to a tremendous diversity in these particularly in the variable region. And the recombination of gene segment has the potential to make around uh, you know uh, 300 different light chains and 600 different, you know, heavy chains that can be produced as a result of this kind of recombination that these are, combined with each other in different patients, and that leads to a different type of peptide or different types of molecule. And during recombination, a further layer of diversity is introduced by enzymes that cut and paste the DNA. So you can say that this is the basic, you know, thing that is going on inside the B cell during this recombination process. You know, there are certain sequences that are present in between these gene segments. For instance, the variable region that you see, this is the lambda chain genes, and the lambda chain genes, and these are the joining uh, uh, sequences of the lambda chain, J lambda, as it's shown here. And you see certain numbers here, 7, 23, 9, 9, 12, and 7. So these, each variable diversity or joining gene segment is flanked by recombination signal sequences. So there are two types of recombination signal sequences. One consists of a nanomer, these nine base pair sequence here and here in purple that you see here. This is a recombination signal sequence, and a haptomer, the seven base pair sequences. Seven is written here and here. These are also recombination signal sequences. And these are separated by 12 base pair sequences. For instance, this is recombination signal sequence or RSS 7, and this is recombination signal sequence 9. And these are, separated by this 12 base pair sequence that you see here. 12 is written here. Sometimes it can be 23 base pair, and sometimes it can be 12 base pair spacer between two recombination signal sequences. And uh, if it is, it can be 12 or 23, but the recombination signal sequences are always these seven base pair and nine base pair sequences that you see here. So recombination occurs only at these spacer sites, different length. What do the recombinases enzymes or the DJ recombinases or enzymes that do recombination? What actually they would do, they would just, cut and paste, these gene segments at various locations and join them together. For instance, it can be cut here at 7, it can be cut here at 7, or it can be cut here at 9, or it can be cut here at this particular position by the enzyme and pasted somewhere else. So these segments are joined together by cutting through these different locations that are shown here uh, in orange and in purple color, the 9 base pair and 7 base pair sequences. A different kind of recombinases are involved in this particular process, and they will lead to making this kind of you know recombination, so that they are recombined with each other, and then that recombination of different segments would lead to this diversity in the variable region. So in the next lecture, I would just tell you that uh, uh, what actually, what kind of further variation is ensured in an antibody, and what is the significance of that with respect to the affinity of the antibody. So thank you very much for this.